you have to know how many fish there are in order to manage. That's integral. And how many fish there are is so many types of science. That's a lot of types of assessment right there.
Advancing scientific understanding of Yukon salmon is one of the Yukon River Panel's major responsibilities and is crucial for improving management.
I've been really lucky to be involved in the Joint Technical Committee. It's a bilateral body, so it's made up of Canadian and US scientists from all backgrounds, all agencies, and it really brings the most qualified individuals, the most diverse scientific backgrounds we can, to come together and look at all of the assessment that occurs in relation to the Yukon fisheries.
The JTC examines data, assesses salmon stocks, reviews management strategies, and provides recommendations to the panel on salmon science and escapement goals. The JTC also helps to evaluate proposals for special research, restoration, and enhancement work funded through the Yukon Panel's R&E Fund.
The JTC is a really awesome body of people that bring together habitat information, experts on population dynamics. We have ocean experts, so it's pretty great because we can all learn from each other. We can all weigh in on methodology and just be making sure that if what we're doing is the best it can be at all times. And so it is a pretty great thing that the Yukon River Panel has at its disposal.
On an annual basis, the JTC is responsible for compiling information on, reviewing, and reporting to the panel the total number of Canadian origin salmon returning to the Yukon River, how many Canadian origin salmon reached the international border, how many Canadian origin salmon were captured in US and Canadian fisheries, and the estimated number of Canadian origin salmon which reached their spawning grounds. Answering these questions is important both for effective management and conservation of Yukon salmon stocks, and for meeting obligations for reporting under the Pacific Salmon Treaty. To help answer these questions, the JTC is relying on new approaches to monitoring and assessment made possible by modern technology. The question of how many fish are making it to the Canadian border compared to preseason predictions can be answered in part using sonar monitoring technology. In the past, the number of salmon making it to Canada was determined mainly through a mark recapture fish wheel program at Bio Island, just past the Canadian border. However, the program was determined to underestimate the number of salmon. After 2008, the Yukon River Panel transitioned to the use of sonar technology at Eagle, Alaska, which has greatly improved our ability to estimate how many Yukon River salmon cross the Canadian border. This information is complemented by a similar program located near the mouth of the Yukon River, near Pilot Station, Alaska, which is used to estimate the total number of Yukon River salmon returning to the watershed annually, and estimate the Canadian origin component of the total Chinook and fall chum salmon runs in season. Sonar sends sound waves down into the water, where they bounce off of objects, including fish, to create a picture of objects in the water, almost like an ultrasound of a baby in utero. By looking at this picture, fish are identified and counted as they pass through the sonar beam. However, sonar cannot consistently identify what species of fish has passed through the beam. The proportion of fish species caught in sonar test fisheries is then used to estimate the number of fish by species. This information can help managers to understand whether the salmon run is smaller or larger than suggested by preseason predictions, and can inform adjustments to in-season management.
While sonars are complicated, they're absolutely the best tool that we have today for counting salmon when we're dealing with a river that's visually occluded. And by that I mean it's silty, it's massive. We can't just look and count the fish. It's really the only tool we have for counting fish abundance.
The question of which salmon are caught in fisheries can be answered through genetic testing of salmon fin clips collected through field monitoring programs. By running genetic tests on these fin clips in a lab, scientists may be able to identify which river system, drainage, or subdrainage a salmon is returning to long before that salmon actually reaches its spawning grounds. On the Yukon River, genetics are used to estimate the proportion of Canadian origin salmon co-migrating with U.S. stocks through the Alaskan portion of the watershed. This information, coupled with harvest surveys, can also be used to provide an estimate of how many Canadian origin salmon are being caught in Alaskan fisheries. But genetics can only be used in this way if we have a genetic fingerprint or baseline of salmon from specific places to compare new samples to. In the past, scientists relied on rough genetic fingerprints that were only able to tell whether salmon were of Alaskan or Canadian origin. Recent projects supported by the Yukon River Panel are creating more detailed genetic fingerprints to more accurately pinpoint where salmon come from. This information can help managers better understand which individual populations of salmon are being harvested in fisheries and inform fishery management strategies. Less than 10 years ago, fisheries managers had much less information available to help them make decisions than they do today. These important scientific developments have dramatically advanced our knowledge about Yukon River salmon and have made it possible to manage fisheries at finer scales. In practice, this has helped to provide more opportunities for sustainable fishing than would have been available with less information and greater uncertainty.